Lettuce now grows real beef protein inside its leaves.
The way we make meat carries a heavy and well-documented cost. Livestock farming demands vast stretches of valuable land to grow animal fodder. It consumes enormous quantities of fresh water, and it pumps methane and nitrous oxide into the atmosphere, accelerating global warming. For many consumers, the desire to prevent animal suffering adds a powerful ethical dimension to these environmental pressures. These forces have already reshaped grocery store aisles, fueling a global market for meat alternatives now worth between 6.7 and 8.1 billion euros per year. That market is projected to keep growing at a rapid clip, somewhere between 8 and 12% annually over the next decade. Until now, the dominant high-tech method for creating the taste of meat without the animal has been microbial engineering. Companies take genes that code for animal proteins and insert them into the genomes of bacteria or yeast. Those microbes are then grown inside massive steel bioreactors, a process known as precision fermentation. But a team of scientists has just proven a radically different approach is possible, one that replaces the steel tank with a living leaf.
Scientists at Imperial College London have for the first time stably produced a real animal protein inside the leaves of ordinary higher plants. The protein is myoglobin, a heme protein found in the heart and skeletal muscle of vertebrates. Myoglobin is rich in iron, which gives meat its distinctive metallic and umami flavor, and it binds oxygen, which delivers the red color consumers associate with a fresh cut of beef. The research team, led by Dr. Alexia Groff, cloned the genes for both pig and cattle myoglobin. They then used a device called a gene gun to physically shoot optimized copies of these animal genes into plant seedlings. The target was not the plant's main genome in its cell nucleus, but the separate, ring-like genome inside its chloroplasts, the tiny compartments that conduct photosynthesis. The plants they transformed were tobacco, a well-established model organism for this kind of technology, and lettuce, an edible crop that could eventually be used directly for food ingredient production. The transformed seedlings grew to full adulthood, flowered, and set seed. Tests confirmed that the next generation of plants inherited the myoglobin-producing trait. The plants remained healthy and fertile, and their photosynthesis showed no significant impairment. Measurements revealed that the lettuce accumulated roughly 810 milligrams of myoglobin per kilogram of dry weight. The tobacco plants produced a nearly identical amount, around 800 milligrams per kilogram. This yield is considerably less than the concentration of myoglobin found in a piece of animal meat, but the environmental arithmetic changes the equation. The researchers also ran a parallel experiment, inserting the same gene into the chloroplast of a unicellular alga and into the much larger nuclear genome of both plant species to compare the efficiency of different production sites.
This breakthrough means that a head of lettuce or a field of tobacco can now function as a miniature protein farm, growing a core meat compound right inside its leaves. For an ordinary person, this changes what the word plant-based can actually mean. A burger made with these proteins would not rely on a synthetic approximation of meatiness. It would contain the very same molecule that makes a beef patty taste metallic and savory, and look red before it hits the heat. The protein is not an imitation. It is the real animal compound, simply produced by a plant instead of a cow. This could directly address one of the most stubborn barriers to the adoption of meat alternatives, which is the simple fact that people all over the world continue to eat meat because of deep-seated taste preferences, cultural habits, and the nutritional benefits it provides. A plant that already makes the flavor and color of meat inside its cells could narrow the gap between a conventional burger and a sustainable one. It also shifts the image of the technology from a factory floor process to an agricultural one, a field of green plants rather than a warehouse of steel tanks. The lettuce in a sandwich or a wrap could one day carry a protein profile that is closer to a complete meal, grown not in a lab but in soil and sunlight.
The winners in this shift could be numerous. Consumers could gain access to meat alternatives that taste more authentic and carry a lower environmental footprint, without the high energy demands of a pharmaceutical-style bioreactor. Farmers could eventually cultivate a new class of high-value crops that produce specialty proteins for the food industry, using existing agricultural land and know-how. The planet stands to gain if this technology helps displace even a fraction of conventional livestock farming, with its heavy toll on land, water, and the climate. The losers are less immediately obvious, but no less real. The precision fermentation industry has poured massive investment into building bioreactor capacity, and a competitive agricultural method that scales at a fraction of the cost could strand those assets. A recent life cycle assessment from the University of California, Davis, found that lab-grown meat using current pharmaceutical-grade production methods has a global warming potential 4 to 25 times greater than retail beef. If plant-based molecular farming proves cleaner and cheaper, it could undercut not just traditional meat, but also the first wave of high-tech alternatives. The scale of what is at stake is measured in the billions of euros of the alternative protein market and the projected growth of global protein demand as the human population climbs toward 10 billion people.
The facts together signal a non-obvious pivot in the entire alternative protein sector. The real race is no longer just about making animal proteins without animals. It is about finding the cheapest, most scalable host for those proteins, and the chloroplast of a plant has just entered the competition as a dark horse. The key insight is about the production platform itself. Precision fermentation requires a sterile, energy-intensive steel bioreactor, a supply chain of purified growth media, and a factory to house it all. A plant leaf is a self-assembling, solar-powered bioreactor that builds itself from soil, water, and carbon dioxide. The Imperial College team targeted the chloroplast specifically because of its bacterial ancestry and its high copy number per cell, which makes it generally much better at churning out large amounts of protein than the cell nucleus. This is why the yield, while lower than an animal muscle, is a starting point, not a ceiling. The why now is written in the convergence of market pressure and scientific capability. The alternative protein market is growing at a double-digit rate just as the limits of fermentation capacity are becoming clear. Motif Food Works, a company that currently makes myoglobin via precision fermentation, has publicly stated that the number of companies leveraging this approach now outnumbers the available production capacity. That is why Motif has already partnered with a molecular farming startup called Ingredient Works to explore whether corn can serve as an effective host for myoglobin production. The Imperial College is an isolated curiosity. That is the story. If this made it clearer, subscribe and turn on notifications so you never miss what happens next.